Swayam Prabha. Digital India. Educated India.
Hello and welcome to today's lecture of Introduction to Mechanobiology. So, over the last few lectures I have been discussing about nucleus, what dictates the nuclear properties, and how forces get transmitted to the nucleus. So, in this regard, I specifically discussed about lamins. Okay. You have A type lamins and B type lamins, and what has been observed in literature is that A to B ratio is tissue specific or tissue stiffness specific. Okay. So, stiffer tissues have A to B ratio is high, and softer tissues A to B ratio is low. Okay. Now, this A to B ratio, so I had also discussed that what is the implication of A to B ratio on motility, okay, or confined migration. Okay. So, in this, so what we observed was, so A to B ratio, so once again A operates as a dashpot, B operates as a spring, okay, and lamin A, so lamin A or lamin AC, because it is a splice variant, gets phosphorylated on softer substrates, okay, and this phosphorylation leads to degradation of the protein, making the nucleus soft. Okay. So, in the context of cancers, okay, what has been observed is that for cancers to invade, you must have mechanisms by which the cell can pass through small pores in the matrix. Okay. And cells cannot deform. So, cells, we have the nuclear deformation limit, which is 10% of undeformed size. Okay. So, the question is, how will then cancer cells migrate? Okay. So, what has been observed is cancer cells, okay, so they down regulate expression of link proteins, okay, including lamin AC, SUN proteins, cash proteins, so on and so forth, nesprins. Okay. But I have also discussed this paper where if the nucleus is too soft, okay, your confined migration can lead to apoptosis. Okay. So, there has been another finding that if you compare the mutation rate of different cancers, okay, what has been found that cancers which originate in stiffer tissues like bone have higher rates of mutation. Okay. So, how can we put the pieces of the puzzle together? So, you have mutation rate increases with stiffness. Okay. So, your laminacy and nuclear stiffness are also positively related, and so if you have nuclear stiffness and migration efficiency, you have a negative correlation. So, if the nucleus is very stiff, then it is less efficient in migration. And not just this connection, you also have higher apoptosis, okay, with increased nuclear stiffness, increased amount of confinement, and when laminacy levels are low. Sorry, this is the other way around. Okay. So, no, this is right only. This is right only. Okay. So, if you have low laminacy levels, you have higher amount of apoptosis. So, is there a link there? Is it possible that when you have migration, confined migration, can this lead to nuclear damage, and somehow this translate into defects, okay, nuclear damage and DNA damage, and can it lead to defects in DNA repair? Okay. So, this is what I am going to talk about today. So, there are two groups, okay. The groups of Jan Lammerding and Matthew Piel, okay, at Institute Curie, and he is at Cornell. Okay. What they showed, that when you have confined migration, okay, when you have confined migration, you can have lot of nuclear rupture events. Okay. How did they prove it? So, what they did, so they created these patterns, okay, these microfabricated stamps, okay. So, imagine you have an EGF gradient in this direction. You have these microfabricated substrates and you have a cell, okay. Let me draw the cell in a different way, okay, which is trying to move in the direction of the chemokine. Okay. Since EGF gradient is high, these cells will overall try to go in that direction. So, what they did was they transfected these cells, they transfected these cells with GFP NLS. So, now NLS is nuclear localization signal. Okay. So, this should ideally only be presented in the nucleus. Okay. But what they found was, during the migration process, every once in a while they would have a situation somewhat like this, okay, where you have the GFP signal would be present in the cytoplasm. Okay. So, it is present in the cytoplasm. Okay. So, when they tracked, so this is as a function of time, they tracked NLS GFP fluorescence, okay, in the nucleus and in the cytoplasm. So, ideally it should be, so let us say this is the nucleus signal. Ideally this should be constant, okay, if the nuclear was never ruptured. But what they found was every once in a while they had a dip and then it returned to this value. Again a dip and a return to the value. And every time the nucleus signal ended up dipping, they found a corresponding increase in the cytoplasm signal, which again fell back to 0, again an increase in the cytoplasm signal. Okay. So, this is your nucleus signal and this is the cytoplasm signal, okay, suggesting that this every time there is a nuclear rupture. Okay. So, since GFP NLS is soluble, so this oozes out into the cytoplasm. Okay. So, this event is a nuclear rupture event. So, this proved that confined migration can indeed lead to nuclear rupture events. Okay. And while this particular geometry was microfabricated, but they have done in multiple collagen gels, and what they found, so if you find out the average pore size, okay, and you find the percentage of cells which exhibit nuclear rupture, they found a correlation. Okay. So, increase in pore size, there is negligible number of cells which exhibit this nuclear rupture. Okay. But if you reduce the pore size, so this is more amount of confinement, more extent of confinement. Okay. More the confinement, more amount of nuclear rupture. Okay. So, however, this rupture does not continue for long. Okay. So, eventually cells restore nuclear envelope integrity using components of the endosomal sorting complexes. So, you have this family of agents called ESCRT3. Okay. So, these are required for transport. Okay. So, ESCRT mediates, again resealing of the nuclear membrane. Okay. So, together what these studies show, that cell migration incurs substantial physical stress on the nuclear envelope, and so if this is true, then you do require efficient nuclear envelope repair, okay, for cell survival. Okay. So, this is the first part. Okay. This shows that you can have nuclear damage, okay, and this would lead to cell death if the cell did not have a mechanism of surviving. Okay. The next part of it is, question is, can these nuclear rupture events lead to alterations in DNA repair? Okay. So, can it be that DNA repair is impacted negatively? Okay. So, to answer this question, Dennis Disher at UPenn, okay, so he used this pore experiment again. Okay. You have these pores, 3 micron or 8 micron pores, okay, from which you transition the cell, okay, you pass the cell through these pores, okay, and during this process, okay, so this is the nucleus which is stretched. Okay. So, they tracked, okay, at multiple points. So, this is my pore, outside the pore, just about inside the pore, and bottom of the pore. Okay. So, what they did was they transfected cells with a multitude of proteins. So, they had GFP H2B, okay, this binds to chromatin. Okay. So, cells were transfected with this, but importantly other DNA repair proteins. Including Q80, Q70, 53 BP1. Okay, so these are all DNA repair proteins. Okay, so what they found. Okay, so again, if I were to just draw the nucleus, let's say this is my nucleus. Okay, they were tracking outside, inside, and bottom intensity of these proteins. Okay. So, what they found, okay, for histone, okay, histone 2B. So, in the 3 micron pore, okay, so they could also track the DNA, right, DNA and the protein. Okay. So, let us say the DNA is giving a given ratio, intensity of inside to outside. So, you are calculating the inside to outside ratio. Okay. If this is the profile of the DNA, okay, H2B give the exact same profile. Okay. Your pore size is 3 microns. Okay. But when you track it, okay, when you track it for, let us say the DNA Q80, Q70 and 53 BP1, okay, for these proteins, this is lot less. Okay. So, suggesting that there is a segregation of mobile proteins, okay, away from chromatin. Okay. So, there is a segregation. So, if I were to draw, okay, the mobile protein fraction, okay, outside, inside and bottom. Okay. In terms of intensity, what they found, if the outside is plus plus plus, the inside is plus and the bottom is plus plus. So, you have in the nucleus, you have a lot here, minuscule here and somewhat more here. Okay. So, versus if you do the same thing for the DNA, okay, the DNA exhibited the reverse thing. Okay. DNA exhibits maximum intensity inside the pore. Okay. And every once in a while, like the previous study which showed that there is rupture, if you tracked the concentration in the cytoplasm, in the bottom there was some signal in the cytoplasm also, suggesting, so this was the nuclear rupture as demonstrated with the other study. Okay. So, this suggests, so there is segregation of mobile proteins away from protein, and this segregation happens at entry and exit of pore, at entry to pore and exit of pore. Okay. So, they then did micropipette aspiration. Okay. Once again they had the DNA labeled and 53 BP1 they labeled, and what they did was, if you have the nucleus, they put the micropipette and they aspirated the nuclei. Okay. And after aspiration, okay, so they aspirated for 1 minute, but then they held the pressure constant for 1 hour. Okay. And what they found was, okay, after 1 hour, okay, if this was the nucleus outline, okay, so most of the DNA was inside, okay, but okay, most of the 53 BP1 was outside. Okay, so DNA was inside the micropipette, but 53 BP1 was outside. Okay, so thus this again proves that there is a segregation. Okay, so they then did this routinely using pipettes of varying diameters. Okay, and what they found, so our x axis is the pipette diameter. Okay. And then what they are tracking here, okay, so what they tracked here was the ratio inside to the ratio outside. So they are tracking the ratio I by O. Okay, so they are tracking the ratio I by O. Okay. So what they found was, with increase in diameter, okay, so this is the DNA intensity ratio, and this is your all the mobile proteins. Okay, they exert completely opposite effects. Okay, so when they considered the mobile proteins, they had repair proteins, repair factors, transcription associate proteins, transcription factors, and epigenetic factors. Okay, as well as nuclease. For all this, what they find, okay, that there is segregation, and this segregation is sensitive to pipette diameter. Okay. Segregation is sensitive to pipette diameter. Okay. In this zone, when you are reducing the pipette diameter, then you see that the mobile intensity inside to outside drops significantly. Okay. So, this is the I by O ratio. Okay. So, this suggests that this segregation, okay, can be a cause of DNA damage, okay, long term. Okay. So, to answer this question, okay, so to answer this question, what they did, say they went one step further. Okay, and what they did was they tracked, so they took U20S, so these are osteosarcoma cells. Okay, they passed them through the 3 micron pores, okay, and then they tracked what is the extent of DNA damage. Okay. So, what they found, so they tracked the extent of DNA damage by the number of gamma H2AX foci. Okay. What they found was in 3 micron ports, whatever be the gamma is, so they had a solid ratio of gamma H2X foci, and what they did was they tracked they found the ratio at the top versus and the bottom. So, they tracked bottom versus top. Okay. So, you have this is the pore, the cell is passing through it, okay. This is your top, this is your bottom, and then they take the number of gamma H2X foci in top, cells which are at the top and cells which are at the bottom, and they find that okay, this ratio in 8 micron pores okay, should be 1, right, because in 8 micron pores the nuclei does not need to be squeezed, but in 3 micron pores there is a nearly 60% increased. So, this is completely migration induced. DNA damage. Okay. So, and this they also observed in other cells like mesenchymal stem cells. What they then demonstrated okay, that the DNA repair, so they did experiments with DNA repair factors, and then they showed that repair factors mislocalize with DNA damage levels. Okay. So, more amount of mislocalization, okay, then you would find that the number of foci will increase, okay, and these changes okay, led to changes in chromosome copy number. And eventually okay, led to genomic heterogeneity. Okay. So, this is a clear demonstration that physical damage to the nucleus can induce heterogeneity. Okay. So, this is an example of migration induced heterogeneity. So, you know, for example, in most cancer cells there are multiple subpopulations which are present. Okay. So, these cells are innately heterogeneous. It is possible that physical factors like pressure can induce compressive stresses, can induce heterogeneity through nuclear damage. Okay. And so, this I would wrap up by saying that in the nucleus okay, you have its physical properties which dictate the migration efficiency and also indirectly okay, they are implicated in heterogeneity. So, a stiffer nuclei, if cells are stiff, cells or nuclei are stiff, then this population should be more homogeneous, okay, less likely to migrate and will be more homogeneous, because this kind of defects will be less likely. Okay. With that, I stop here. As reading assignment, I suggest you to read the following 2 papers. Okay. Thank you for your attention.